To understand the behavior of chromosomes in mitosis, we need to understand what does a chromosome mean. We need to know the structure of chromosome. If we take an example of an animal cell, this is for instance an animal cell. You already know there is a nucleus inside this animal cell. It has a pair of centrioles. And if you observe this in undividing cell under undividing cell under an ordinary microscope, you will see thin threads of DNA and proteins inside it, which is known as chromatin. Okay. So chromatin is thin threads of DNA and proteins known as histones. DNA is acidic. This is acidic in nature. So the basic proteins can easily be associated with it. So these basic proteins associated with it, basic proteins are histones. And this is an acidic DNA. So basic proteins are associated with the acidic DNA. When this cell undergoes division, this chromatin coils and condenses and becomes visible as thick threads that are known as chromosomes. So chromatin coils and condenses during division of a cell and becomes visible as thick threads of DNA and histones. These thick threads of DNA and histones are known as chromosomes. Chromosomes may be visible in a dividing cell as two arm-like structures that are attached with a central disc. These arm-like structures are known as chromatid. So this is an arm-like structure. And the central disc is known as centromere. A chromosome also contains shoelaces tip-like structures at the ends of these chromatids, which are known as telomeres. Before a cell begins to divide, the chromosomes have these duplicated chromatids. So each chromatid contains one DNA molecule. Each chromatid contains one DNA molecule. So as there are two chromatids, there are two DNA molecules found in this chromosome. These two chromatids are known as sister chromatids. And these are identical chromatids. These are identical chromatids. Identical chromatids. Before a cell begins to divide, the DNA is duplicated and each chromosome has two chromatids. During cell division, the centromeres duplicate and these two chromatids are pulled apart. So this centromeres are duplicated and these chromatids are separated and pulled apart. So that they are separated and move to each cell. So each cell will now have one chromatid from each chromosome. These cells will undergo a process of mitosis again. And before it undergoes cell division again, it passes through a phase which is known as an interphase, where DNA is duplicated again. And each chromosome will have duplicated chromatids again. And this process continues during a mitotic cell cycle. The number of chromosomes in each species is specific. is specific in each species. In human, it is 46. In chimpanzees, it is 48. So the different species have their specific number. During cell division, in growth and repair of the tissues, this number of chromosomes that stays constant generation after generation. As we discussed earlier, that a zygote divides into two cells, which are identical to each other, and then it divides into 4 cells. These 4 cells will be genetically identical to each other again. So if there is 46 chromosomes in this parent cell, the 2 daughter cells are made. Each will have 46 chromosomes in them. So you see, there is an additional 46 chromosomes that come from the duplication that takes place during interphase. So before every cell division, the DNA undergoes duplication, and so a chromosome will change from a single chromatid to a double chromatid after duplication. And then after mitosis, each chromosome will have 1 chromatid instead of 2 chromatids.